Right. Welcome, guys. So we are going straight to the industrial and the microbiology and the microbial ecology from what we just studied about immunological studies. So we are going to study how it's going to progress into the industrial microbiology and the microbial ecology. So let's start. Although humans had unknowingly exploited microbes for the thousands of years, industrial microbiology developed in large parts from the work of the yeast Pasteur and the alcoholic fermentation that yielded wine and other alcoholic beverages. Now you guys know why the yeast Pasteur was important, because till now what we were studying in this chapter, most of the work was, one and the other proposed that yeast cells were responsible for the conversion of sugars to alcohol. The leading chemists of that time believed that the microorganisms were not involved. Yeah, it was the first time in 1837 where the Theodore Schwann and others proposed that the yeast cell, the microorganisms, are responsible for the fermentation, aka sugars to alcohol. Before that, the chemists didn't believe that microorganisms could be involved. Like, they were chemists, so of course they were thinking on it from the angle of chemicals. They were convinced that the fermentation was due to the chemical instability that degraded the sugar to alcohol. See, that was their concept. The chemical instability. Pasteur didn't agree. He believed that the fermentation were carried out by living organisms. Well, I don't think it was just a belief. It was belief plus experiments, because science doesn't go with the beliefs, they go with the experiments. In 1856, the why M. Bigot, an industrialist in Lille, France, where the Pasteur worked, requested Pasteur's assistance. His business produced ethanol from the fermentation of the beet sugar, and the alcohol yields had recently declined and the product had been sour. So now that knew that the Pasteur was working with the microorganisms, so people started thinking about things from that perspective, and so he asked for Pasteur's assistance in his fermentation of beet sugar. Guys, this is how beet sugar look like. Wait. Maybe people mostly know it, but usually they don't know. Like, this is how it looks, and it is red in color. Is there a photo of it being cut? Wait, it's a carrot. It is not a beet sugar. Wait, I thought it was not carrot. Oh, beetroot. Look, looks like I was okay. I was talking about this red beet sugar. Alright. I was talking about this red one. Looks like he was making ethanol from this beet sugar, and his work was declining, so he asked for Pasteur's assistance. And okay, Pasteur discovered that the fermentation was failing because the yeast normally responsible for the alcohol fermentation had been replaced by the microorganisms that produced lactic acid rather than the ethanol. Okay, so there was contamination by microorganisms in his fermentation, and they were producing lactic acid, which he called the sour taste, which wasn't from ethanol. So in solving this practical problem, Pasteur demonstrated, like, how was he going to convince the person that asked for him that his work was like going wrong. So this that all the fermentation were due to the activities of specific yeast and the bacteria. He published several papers on the fermentation between 1850 to 1860. Like, there are several ways to get rid of the microorganisms that you don't want in your fermentations, like pH regulation or things like yeast has different pH potentials, and the humidity and things like that, and the bacteria has others, so you can get rid of the contamination from your fermentation. So I think he might have given the idea of that to the person that was doing the fermentation. After that, he published several papers on the fermentation between 1857 to 1860. His success led to a study of wine diseases. Wine diseases, guys. Like, I think might be people drinking wine and they were getting diseases. How was he going to study that? So, and develop, he studied the wine diseases, and he developed the pasteurization to preserve wine during the storage. Okay, so the diseases was going to spread, like was spreading by the wine, so he developed pasteurization method to preserve wine, so there is no contamination of microorganisms in it, and so no further diseases. And in our case, the microbiology, this is not disease, this is infection. Pasteur studied on the fermentation continued for almost 20 years. 20 years he was studying the fermentation. That's some kind of, dedication to something. One of his most important discoveries was that some fermentative microorganisms were anaerobic and could live only in the absence of oxygen, whereas others were able to live either aerobically or anaerobically. Of course, after working with fermentation for 20 years, these kind of things, these things are, normal to come across. So they are saying controlling food spoilage. We are going to read in the next chapter, or not next, but the future chapters. So this was the industrial microbiology. So industrial microbiology is usually, I think, based on the fermentation processes, like the whole industry. It is just you using the microorganisms and making the products you desire by either changing the microorganisms genetic material, or by just simply using their machinery to produce their products and change their material. So again, it started with Pasteur and ended with Pasteur, because pasteurization and fermentation were his expertise. So in this paragraph there is nothing written more about the scientist. Now, microbial ecology developed when a few of the early microbiologists chose to investigate the ecological role of microorganisms. Alright, so we all know that the microorganisms are here and there and everywhere. So the microbiologists thought, why not go and search about the microorganisms that are present in our environment? So in particular, they studied the microbial involvement in the carbon, nitrogen, and sulfur cycle taking place in the soil and aquatic habitats. Oh, right. So they didn't just took the samples and did something here and there. They were already considering the cycles, like if there is any role of microorganisms in these cycles. The Russian microbiologist Sergei Vinogredsky made many contributions to soil microbiology. Let's read its name again. Sergei Vinogredsky. Sergei Vinogredsky. So he contributed to the soil microbiology. How? He discovered that the soil bacteria could oxidize iron, sulfur, and ammonia to obtain energy. Alright, so the bacteria that he was talking about, it was saprophytic, right? They were eating on the almost minerals, I think. Sulfur. Like there are sulfur bacteria. I don't know about the iron ones. Ammonia, yeah, ammonia, mostly bacteria used in the soil for their energy. So in case of ammonia, it is involved in the nitrogen cycle. In case of sulfur, it is involved in the sulfur cycle. I still don't know what is oxidizing iron could do, but yeah, they could do it. And the many bacteria could incorporate carbon dioxide into organic matter, much like a photosynthetic organisms do. Like they discovered that there were bacteria that acted like plants, like that could make food. Not the algae, not the fungi, we are talking about the bacteria. And the cyanobacteria, which uses the carbon dioxide of the air and convert it into oxygen, I think it's a good example of a photosynthetic bacteria, and it was originally there when the world had no other living organisms on it. Vinogredsky also isolated anaerobic nitrogen fixing soil bacteria and studied the decomposition of the cellulose. So of course, when he was studying about the soil microbiology, and this is just a so new era, so there are so many microbes in the soil, so he was discovering it one after another. So he isolated the anaerobic nitrogen fixing soil bacteria. Well, they are anaerobic because they are under the soils, and the further you go down, the further they don't have the oxygen and they survive here and that. So that's why they are anaerobic, and they live in the roots of the plants. So the nitrogen air, like the nitrogen gas is fixed by them, converted into ammonia, and the ammonia is converted into nitrates, and nitrates, like plants can take the nitrogen directly from the air. They have to have it in a tangible form, like, sorry, the gas is also a tangible form, like in the nitrates or nitrites form. There are fungi, I think, that uses the ammonia, or maybe some plants, very little rare plants that uses the ammonia. Alright, they study the decomposition of cellulose, decomposition, degradation, or decomposition. Well, we know the bacteria decompose cellulose as well. Ever heard of the termites? The insects that eat the wood and make it, hollow. Yeah, hollow. So they have the bacteria in their stomach for the digestion of the cellulose. Wait, let's go here. Termites. These guys are involved in the, see, these guys stomach have the bacteria that shows that digests the cellulose. Alright, Martinus Beijerinck. The Beijerinck already did what? The tobacco mosaic virus discovery, right? Was one of the great general microbiologists who made, if you see from the tobacco mosaic angle, that means he was an ecologist as well, like not ecologist but studying the plants, so the plant microorganisms. And they were saying that he was a great general microbiologist, means he wasn't specifically involved in one field of microbiology, so who made fundamental contribution to the microbial ecology and many other fields. He isolated the aerobic nitrogen fixing bacteria. So the Graysky, So the Graysky discovered the anaerobic nitrogen fixing bacteria, while the Beijerinck discovered the aerobic nitrogen fixing bacteria, and that was the Azobacter, a root nodule bacterium. Guys, this guy is so important in the symbiotic relationship. We almost every time include this Azobacter in the symbiotic relationship. A root nodule bacterium that is also capable of fixing nitrogen, later named Rhizobium. And sulfate reducing bacteria, the bacteria that is involved in the sulfur cycle. In some way or another, we are going to this cycles, I think, in the future days of this book. So Beijerinck and the Winogradsky also developed the enrichment culture technique and use of the selective media, which have been of such great importance in the microbiology. Yeah, that's so true. We use enrichment culture techniques. Enrichment culture techniques is, you have certain media, right? It has normal amount of nutrients in it, but when we talk about enrichment, we increase the amount of a single nutrient or two nutrients, like which separates it from the other nutrient medias. Like for example, we add salt, a lot of salt in a media, and it will only grow the bacteria that could resist this high amount of salt, also known as halophiles, the ones that could live in the salt environment. So this is one type of enrichment culture media, right, technique. Like there can be sugar, like a lot of sugar you add, and that becomes the enrichment culture technique. While there is a use of selective media. Selective media is the one that can grow certain bacteria and cannot grow certain bacteria. Like, there is a media that is selective, and there is one bacteria that is differential. Selective media versus differential media. Guys, this is very important questions always asked in the microbiology. Um, should we study from here? No, we can't see it. Selective media allows, wait, let me enlarge it a little bit. I need to make a shortcut of this. Can't go back every time to change it. Selective media allows certain organisms to grow while preventing others from growing, while differential media helps distinguish between the organisms based on their biochemical properties. Yeah, like selective media is like you separating the bacteria and the fungi from one another. Like the bacteria would need nutrient agar and the fungi would need the if it is a lactose fermenter or if it is not a lactose fermenter. See, the mannitol salt agar, the tyrine matin agar. Wait, let's search it because it is used. Salt agar is selective for what? For staphylococci that can tolerate high salt concentration. Alright, when I was talking about the high salt concentration, you can take the example of mannitol salt agar and put it into that. So even in bacteria it is only selective for the, but then wait, what is the differential media? Was I saying it oppositely? Example of differential media. Again, look at this. It's a mannitol salt agar, a selective and differential growth. No. We need to see something different. Right, a selective. I'm talking about just the differential media. It allows the growth of the various microbes which can be differentiated by their method of hemolysis. Wait a second. Alright, so in differential media it is the same media that is used by two organisms, but their way of coping with that media is different. While in selective media they can't even grow on that. Right? Like for example in lactose fermenter differential media, the bacteria that could break down the lactose are, make the pink colonies, while the one that cannot doesn't make the pink colonies. Like they both can grow. It's not like they can't grow, but they cope up with that media in different ways. So that's why it's a differential media. While in selective media, a type of organism is allowed to grow while another type is not allowed to grow at all. Like it cannot grow there in any way. Well, yeah, the example that the fungi and bacteria has different culture is also coming in the selective media. So which have been of such great importance in microbiology. I think as we go with the book, you guys will know how much important is this, because microbiology starts from the culturing techniques, from the media we use. So was this all about the development of industrial microbiology? Alright, so they are just stating about the cornerstones of the industrial microbiology and the microbial ecology. So what we studied here was the industrial microbiology was dealing with the pastures work about the fermentation, and like someone asked, the Mbigo asked for his help, and he found out that the contamination by microorganisms were done and when yeast wasn't working fine. So that was that. While in microbial ecology, people started studying the microorganisms involved in the carbon cycle and nitrogen cycle, etc. While the first one to do that in this advancement in the soil microbiology was the Sergei Winogradsky, and his contribution was like, discovering the bacteria that could oxidize iron, sulfur, and ammonia, while the Martinus Beijering contributed to the aerobic nitrogen fixing bacteria here, like which is called as Azobacter. And then the biggest thing was not discovering the soil bacteria, it was discovering the enrichment culture techniques and the use of selective media. I think the amount of bacteria they discovered was just because they discovered the selective media and the culture techniques. If they didn't have developed these, there was no way for them to obtain all of these different kinds of microorganisms, since they all have different requirements for them to grow. Oh, alright. So now we are going to the scope and relevance of microbiology. Scope and relevance. Okay, we are not going to study this today. This is a long chapter. So I think if you guys want to study the real history of microbiology, I think it's better to watch the previous videos about this history. Like you are going to study the microbial discoveries along the timeline with the natural world that existed at that time with physics, chemistry, or wars and things like that. So how much is left in this chapter? Look at them, their haircut. The future of microbiology. Alright, we have just 2 topics left. So before going to the next chapter, we are going to cover these critical thinking questions and these type of questions that came, in the middle of them. Like this is important. It's not like we are going to just move forward and forget everything we studied. Alright, that was it.